I'll begin by acknowledging that we meet today on the traditional lands of the Wurundjeri people of the Kulin Nation, and we pay our respects to their elders past, present, and emerging, as well as other members of the Aboriginal and Torres Strait Islander community that are here with us today, or wherever you may be joining us online. So it's always a bittersweet moment in a supervisor's time when their student is presenting their PhD oration. On the one hand, extremely proud, especially for all the students that have gone through this cohort during the pandemic and the difficulties that you all encountered with managing a PhD during periods of lockdown. Always extremely proud to see a PhD student finish their PhD, but also always sad to see them go. And so we have Pradip Roy, who's presenting today on his work over the last 4 or so years in my lab, understanding aspects of the basic biology of a really important gastrointestinal parasite, which is Ascaris sum. It's an important parasite in pigs, but also a model for a parasite that causes, as you'll hear, infections in around 800 million primarily children around the world. And it's been fantastic to watch Pradip learn in this program and develop. And as you see in the presentation he's about to give, and I won't steal his thunder, he's developed a really fantastic set of skills across quite a broad range of areas, from bioinformatics and transcriptomics, understanding the biology of these parasites, but also developing assays into their behavior and chemosensation. And it's a really satisfying story, and then I'll shut up because it's his moment, to see this program, because this was something that stemmed out of a collaboration that had started more than 10 years ago. This idea that during the early phases of Ascaris infection, it was playing an active role through chemosensing its host, and that was critical to the infection components. And then to see finally years later that program be funded, and then to have a student take it on and have a student not only take it on, but do such an excellent job with it and demonstrate that there is a fundamental misunderstanding in the biology, in the life cycle of a parasite that affects 800 million people around the world. And to see him demonstrate that so well and so beautifully, as you'll see in this presentation, is really satisfying. So I'm really, really proud of the work that Pradip's done. I think he's been an absolutely outstanding student. He's a wonderful person to have in the lab. I'm really proud of the time he spent with us, and I'm really excited to see what he does next. And so with that, I'll move aside because he's the star of the show. Please welcome Pradip to the podium, and we'll see his thesis. Well done.
Thanks, Aaron, for your nice introductions. So today I'm going to talk about my thesis, which is titled as exploring the chemosensation in Ascaris infections. So as Aaron told, Ascaris infections is one of the most prevalent infectious diseases around the world. The disease caused by the Ascaris species is known as the Ascariasis. It infects around 800 million people around the world. The mortality rate is around 60,000 per year. The children are the most vulnerable group of these infections, and apart from the public health concerns, they have a huge impact on the livestock industry also. If you look at the geographic locations of the Ascariasis, it's most common in the warm and the moist heat weather countries, mostly in the tropical and subtropical regions, with poor sanitation and hygiene. And if you look at the transmission of the Ascaris infections, we see that the infections happens due to the contaminated water, or contaminated food, or unwashed hands and the unwashed fruits and vegetables. However, due to the less mortality rates than the morbidity, and due to the lack of disease model, till now, Ascaris is known as the most neglected parasite globally. So if you look at the clinical presentations of the Ascariasis, as I told, children are the most vulnerable group of that infections. It causes malnutrition and reduced physical and cognitive development in children. In case of heavy infections, it causes the stunt growth in children. And in case of high worm burden, it causes the abdominal pain and intestinal obstructions. And in severe cases, it causes death. Apart from that, migrating adult worms in the body, it's caused the biliary tract occlusions, appendicitis, and nasopharyngeal expulsions. So if you look at the current control strategies, if you look at the life cycle of the Ascariasis, we see that when Ascaris eggs are ingested through the host, it directly comes to the intestine, and they hatched into the intestine. After that, immediately they evade the uh, intestinal mucosa, and then like it goes to the liver. And coming to the liver, they directly migrate to the lung, where they like evade the alveoli and come up to the trachea. And after that, they come back to intestine for the second time, and they stay there for like 1 to 2 years to become adults and release eggs to the feces. So this migration is known as like the from intestine to liver to lung is known as the hepatopulmonary migrations. So to control the ascariasis, there are several approaches, including the treatment approaches. The most common one is like the sanitations and the personal hygiene with health educations. But as I told, like this is the endemic in the most underdeveloped countries. It's a really slow process and it's required high cost. Apart from that, like the vaccine might be like good alternate options to treat the or control the ascariasis, but it's not developed yet. And then the epidemiology of the infections is not like well reported, so it's pretty hard to understand the epidemiology of the overall the world accordingly. So the most common treatment approaches for the ascariasis is the like anthelmintic treatments. There are several anthelmintics available in the market, which are like albendazoles, mebendazoles, levomisols, and parenteral palmate, etc. Among them, like single oral dose of benzimidazoles can control the ascariasis, but it's only active on the adult stages of the worms and doesn't protect against the early phase of the infections. So if we come to the current limitations of the anthelmintic treatments, so there are several limitations currently available in the anthelmintics. The most common one is the reinfections. So current anthelmintic doesn't give protection against the reinfections. So it means like when they are being treated with the anthelmintics, the disease infections coming up again, like for Ascaris, after 6 months of the treatment, the chance of getting the reinfection rate is 55, and for who comes is like more than 80%. Apart from the reinfections, there is a possibility, there's a report of the reduced efficacy of several anthelmintics on different nematodes. Like for example, like benzimidazoles shows reduced efficacy for Ascaris infections and or ivermectin for the Onchocerca. Apart from the reinfections and the reduced efficacy, there is a threat of drug resistance due to the mass drug administrations to deworming the population in the endemic regions. However, there is not reported that in human anthelmintics there is a drug resistance, but it has been observed that the drug resistance happens in the livestock anthelmintics. So we have to find out a new intervention to control the Ascaris or parasitic diseases urgency. So most of the anthelmintic drugs available in the market, they target the muscular neurotransmitter or nervous systems of the nematodes. Apart from the benzimidazoles only targets the beta tubulin structure of the nematodes. But this is a great opportunity to develop a like targeting the nervous systems of for the parasite nematodes, because most of the anthelmintic neurotransmitters, if you block them, they paralyze the parasites, and this paralysis, it seems like they can't move, but it's actually they can't make the decisions. So nematodes actually take decision based on their cues or sensations. So if you can block the chemosensations or their migrations using the puzzling their decision making, we can actually control the early infection in the early stages. And we assume that chemosensation may play a vital role in completing their life cycle in the host. So that's come to the point, like what is chemosensations? Like chemosensation is one of the most ubiquitous process in all of the animals. Like it's the animals needs to sense to the external environments for seeking foods, shelters, find mates, or like they avoid the predators. And they senses the chemical stimulus from the environment and they convert it to the neurological system signals. And by this how they works like that to the specific functions. So it's like representation of how they are actually sensing the cues from the environment and act accordingly. But it's in the overall animals. But if we come to the nematodes, what is happening in chemosensation in nematodes. So it is well studied in free living nematodes. It's not coming. It should come. It's coming. So it is well studied in the C. elegans, which is a model organism, and it's a free living nematodes. And in C. elegans, they use both volatile and water soluble cues to find the food. And they have a like simple neural structure, consist of like 302 neurons in total. And among them, only 12 pairs of them are sensory neurons. They control the overall sensory functions in C. elegans. These 12 pairs of sensory genes actually bilaterally symmetrical, but report shows that the functions of these pairs might be different based on the different functions. In C. elegans, most of the chemoreceptors are the G protein coupled receptors. And apart from them, like other downstream signaling are also there, which included like the heterotrimeric G proteins, guanide cyclase, transient receptor potentials, and cyclic nucleotide gated channels. And you can see that list, like each single neurons have their own specific functions. Like for example, the AWC neurons are like functions on the volatile chemotexis included with receptors and other downstream signaling genes. Then we come to the signal transactions, like how do they sense and transmit the signals to the cell membrane. So actually like the sensory stimulants actually sense the molecules from the environment, and then they like received by the GPCS, and they activated the heterotrimeric G proteins, and then they activated the guanylate cyclase, and by this how they opens the CGMP gated channels using the encoded by the TAX and TAX2, TAX4, who are the cyclic nucleotide gated channels, and then they by this how they depolarize the cell. So that's a like process of like how they are transducing the signals using the GPCR cascade into the cell. And these GPCRs regulates the chemosensations, lipid homeostasis, social behavior, etc. in C. elegans. So it is well studied in like the free living nematode C. elegans, but in case of parasitic nematodes, it's not well studied. There is a few studies happens for reported for the host-seeking nematodes. The parasitic worms, those parasitic worms like use the sensory cues to find and infect their host. And this host-seeking involves multiple sensory modalities. And it has been reported that they have a specialized olfactory systems for host finding. But it's still like it's not well annotated. We have to depend on the knowledge of C. elegans chemosensations. And if you look at the heat map, you can see like lots of chemicals are being attracted or repelled by the different skin penetrating nematodes, entomopathogenic nematodes, and the free living nematodes. So it shows that like that the parasitic nematodes have some sort of like chemotexis functions, and they are responding to different chemicals. And what are those chemicals? Like specifically you can see. So like for host seeking, we can see like the host always release some like volatile or soluble chemicals from their body parts, like for skin, sweat, breath, serum, etc. And the nematodes actually, parasitic nematodes, they actually attracted to the skin extract or like host serum or even like the carbon dioxide, which is the respiratory excrete from the body. And the report shows that like for example, urocanic acid is a like major chemoattractant for the strongyloid structural release. So this urocanic acid is the major chemical component of the human skin. So by this how they just like find out the host and invade the host. Apart from the like Nicotin americanus attracted to the sodium chloride, that is a major component of the human sweat or like mammal sweat. So as I told, like it's not well studied in the other parasitic worms. So we are interested to see the chemosensations in Ascaris as a model organism. So even again, if we look at the life cycle of Ascaris, we see that Ascaris larvae, after hatching into the intestine, they go through a complex migratory pathway known as the hepatopulmonary migrations from liver to lung. So a study shows that the genes which have been transcribed during the L3 stage and L4 stage, L3 stage in liver and L3 stage in lung, the genes which are transcribed significantly, they are related with the chemotexis in C. elegans based on the ortholog analysis. So that's give us the idea that this migration process might be like active process guided by some chemical cues from the body or host, apart from it was believed like it's a passive process previously. And based on that, we hypothesized that these hepatopulmonary migrations in Ascaris might be regulating by the chemosensations. And why we took the Ascaris as a model organisms, like as we know, like Ascaris is the large roundoms of pigs, and they have like similarity with the human roundom Ascaris lumbricoides. So in both way, like we can study the same organisms at once, and they are bigger in size, like mostly like 30 to 35 centimeter when they have become adult, and they have a long explored neurobiology study, and their neuron size is like really huge in the adult stage, like more than 5 to 60 micrometer. And their neurological system is like mostly common with the C. elegans, and the draft genome of Ascaris is available, so we can use the molecular biology based on the genomes of Ascaris. So if you like see the like what is the size of the Ascaris swarms, so it's like shows the how big are they, and like you can actually see how they're moving, and like they're really big, huge. You can see that. Anyway, so based on that, like overall that introduction, we assume that chemosensation play a key role in the hepatopulmonary migrations, and if we can block the hepatopulmonary migrations, we can block the early infections of the disease. So to do that, I came to my PhD research aims. So one is like, first one is like the exploring the chemosensory signaling in Ascaris, insight from the comparative genomics and transcriptomics, differential transcriptomic analysis of Ascaris sum larvae stimulated by the pig liver and lung lysate, and the fabrications of microfluidics, and exploring the chemotactic responses of Ascaris sum larvae to pig liver lysate. So if you come to the first one, to explore the chemosensory signaling in Ascaris, so what I did, like, I used the bioinformatic approaches to curate all of the ortholog-based chemosensory genes in Ascaris sum. Then we take the head portions of the Ascaris, adult Ascaris, dissect them into the head and body portions, and then we dissect the head into the left head and right head to see is there any symmetry or asymmetry. And we did the RNA-seq based on all of those specific tissues, and then analyze the differential transcriptomes analysis. So this RNA-seq was done by Joy Liu, who was the honors student in Jake's lab. So as I told, like Ascaris is a non-model organism, and their genome is not oil annotated. We have to depend on the ortholog-based analysis to curate all the chemosensory GPCRs or like the other signaling genes in Ascaris. So we use a hidden mark model to curate all the GPCRs from Ascaris sum, and we characterized all the 5 classes of GPCRs including rhodopsins and other secretin freezols, glutamate, and adhesions. So if we look at the summary of curating GPCRs in Ascaris sum, we saw that Ascaris contains a reduced set of chemosensory GPCRs. So among the rhodopsin, they have a chemosensory GPCRs, which are unique and specific to the nematodes, and that's not found in the other organisms. So that's why we focused on that, like see that chemosensory GPCR is actually reduced in the Ascaris sum. So to confirm that thing, we did the pan-phylum analysis from the other clades of the nematodes, and we saw that it shows the same pattern of the parasitic nematodes actually contains the reduced chemosensory GPCRs compared to their free-living counterparts. It has been also reported by several studies that like the GPCRs, chemosensory GPCRs in nematodes are related with the environmental exposure. So if they are exposed to the broad environment like free-living nematodes, they have more chemosensory GPCRs than the parasitic nematodes, because it might be like C. elegans, they have to expose to a like broad environment and they have to detect lots of cues. On the other hand, parasitic nematodes are host specific. They have to respond to like a specific environment, so they don't need much chemosensory GPCRs. Apart from that, when we did the phylogenetic analysis of those chemosensory GPCRs, we didn't observe any one-to-one ortholog with Ascaris sum to C. elegans or other free-living nematodes. So it shows that actually the Ascaris sum has a diverse set of chemosensory GPCRs that might have like unique functions in them. And also you observe that like in clade 3, which have like Ascaris sum, Toxocara canis, or Onchocerca volvulus, they have like unique set of chemosensory GPCRs which are specific to clade 3. which are highlighted here in the phylogenetic tree. So that's give us the idea like the chemosensory GPCS in Ascaris are really diverse and reduced. Apart from that, we curated all other downstream signaling genes, which are like receptor guanylate cyclase, soluble guanylate cyclase, heterotrimeric G proteins, other ion channels, and most of the downstream signaling genes are showed conservations throughout the nematodes. We only just like Ascaris is missing like soluble guanylate cyclase, and this guanylate cyclase is actually involved in the oxygen sensations. I think Ascaris doesn't like oxygen and they love the low oxygen level environment. So that's why might be with the genomic evolution, they might lose that soluble GCY guanylate cyclase genes. So after that, so we curated all sensory genes, possible sensory genes based on the ortholog analysis. Then we want to see like what is the transcriptomic profiling. So we want to see like that in tissue specific level, where they are enriched in the transcriptomic level. We assume that in the head, the sensory genes should be upregulated or like enriched because it shows that in C. elegans, the sensory genes are being in the head region of the nematodes. So we did the tissue-specific transcriptomes to the left head, right head compared to the body, and we saw that most of the sensory genes are transcribed significantly into the head compared to the body portions. If you look at the heat map, the heat map shows that the genes that are transcribed significantly in the head region, they all are involved in the several chemosensory function in C. elegans, whereas the genes upregulated significantly into the body portions, they are mostly involved in the other physiological functions. And based on our genes enrichment of sensory genes, we see that as a set of sensory genes, they upregulated or enriched towards the left head and the right head compared to the body portions. And then like we do the functional annotation of those significantly transcribed genes, and we see that the genes which are like signaling pathway involved in the G protein coupled receptor activity, or like the neuropeptide signaling pathway, or like ion transportations, or are enriched into the left head and the right head, but not in the body. So that's transcriptomic profiling of this head versus body shows that Ascaris has a like compact and robust chemosensory system. So to summarize that, so we see that Ascaris has a reduced and diverse set of chemosensory GPCS. They have a conserved downstream signaling genes, and the sensory genes and signaling pathway enriched into the head. And we didn't observe like much differences in the left hand and right hand transcriptomic profiling, and that shows that they have an active chemosensory systems. Apart from that, we did a small like immunohistochemistry analysis to see the neuronal structure of Ascaris head, and we detect and see the neurons are positioning into the head portions of the Ascaris. But though I'm not talking about this thing today, but we see that. So that means shows that the genes are being there, chemosensory genes, and the neuronal structure are there. So they have like active chemosensory systems. But then what next? What happens when they sense the liver, host liver and lung? Are there any signaling pathway coming up or not? Or is there any chemotricity genes are coming up or not? So to do that experiment, we come to our second aim. So to analyze the differential transcriptomic analysis of Ascaris larvae stimulated by the pig's liver and lung homogeneous. So to do that, we have developed an agar migration assay, which is developed by Luis in a lab. So that's basically a small experiment which I use for the enzymatic screening of nematodes. So for doing that, we actually took the freshly slaughtered pig's liver and lung, and then take the homogeneous from the liver and lung extract and did the migration assay, putting the Ascaris larvae into the loose 1% agar and put the liver and lung extract, see how they are migrating towards the liver and lung homogeneous compared to the RPMI as a control. So we see, like interestingly, the number of larvae that are being migrated towards the liver extract is significantly higher than the number of the larvae which are migrated to the lung extract, or RPMI. So based on that experiment, we concluded that these preferential migrations of the larvae towards the liver extract might be due to the presence of specific liver cues, and which has been detected by the larvae. So that's give us an idea, like they have some sort of like chemosensory functions, chemosensory attractions. Then we did the differential transcriptomics of those migrated larvae, which are migrated from the RPMI, lung, and liver extract, and did the differential transcriptome. So we saw that like genes are differentially transcribed when they are exposed to liver and lung extract. And the common gene go terms related with the migrated larvae in the both cases are the ribosomal biogenesis, RNA processing, locomotions, and other like physiological functions. So as we saw that the larvae are more migratory towards the liver stimulated larvae. So next to the sections, we just focused on the liver stimulated larvae. What is happening there? Is there anything coming up like related to the chemotexis or any signaling pathway is being coming up? So when we check only specific or unique genes transcribed significantly in the liver stimulated larvae, we saw that the fatty acid metabolism related genes, the cilium morphogenesis genes, neuropeptide signaling, and chemosensory signaling genes are being upregulated. So fatty acid metabolism is one of the crucial methods for the nematodes for the survival and health. And as we know, liver is like and this is the loss of fat. The Ascaris larvae might be taking the liver homogeneous as a like active source of food or nutrition, as they need the energy to go through that complex migratory pathway from the liver to lung and then come back to intestine. So they need the energy. So that might be like they're taking or detecting as a food source. So that also gave us an idea like that, okay, they are using that liver as a nutrient source or something. So then like we need to see like that these migrations is like regulated by some other chemosensory related functions or not. So we saw that like cilium morphogenesis as I told, and the chemotexis related genes are coming up, and the transcribes differentially and in the liver stimulated larvae, and it's like cilia is the one of the most major component of the nematodes to sense different sort of like food, chemicals, or mating or temperatures. And we found like in Ascaris, there are several genes are coming up which are related to the cilium morphogenesis, which are involved in the interflagellar transportations, cilium assembly, and any defect of any single of genes in C ligands, they showed that they shows the defection in the chemotexis. So that means like it shows that the chemotexis is actively regulating when they are detecting the or stimulated by the liver homogeneous. Apart from that, like there are other chemotexis related genes are also upregulated in including heterotrimeric G proteins, guanylate cyclase, or cyclic nucleotide gated channels are also being upregulated in the liver stimulated larvae. So then like we ask questions like what signaling pathway like is being coming up like regulating that process like migrating? Is there any signaling happenings from the liver that like they are being provoked to get that signal? So we saw that in our data set, like in transcriptions, we saw that several neuropeptides and neuropeptide GPCS are transcribed significantly. So that's why we are interested to see what is happening in the neuropeptide signaling in Ascaris in the liver stimulated larvae. So neuropeptides are actually everyone knows like it's small peptides, and we just focus on the two types of neuropeptides like afaramide like neuropeptides or neuropeptide like peptides, and they are actually involved in the like lipid metabolisms or lipid detections or fatty acid metabolisms regulations by these two neuropeptide signaling. So based on that, we identified like 26 FLPs and 51 NLPs in Ascaris based on the C elegans, and then we saw that 8 afaramide-like peptides or 12 neuropeptide-like peptides are significantly transcribed in our dataset in liver stimulated larvae. And apart from that, like neuropeptide processing genes are also upregulated, which are the important genes to make the neuropeptides from the neuropeptides encoding genes. And they have also, like we saw that, like as I told, neuropeptide GPCS, which are the receptor for the ligand, are also coming up. So it shows like the ligand as like peptides and their receptors are like upregulating significantly. We assume that during that hepatometabolic migrations, the neuropeptide signaling are involved in this process. And also we saw in our data set, which are significantly transcribing the liver-stimulated larvae, we saw several nuclear hormone receptors are coming up. But these nuclear hormone receptors are actually shown that in C. elegans, they are involved in responding to the environmental stimulant. So that's why we are interested to see that, because as I saw, like 4 of the neuropeptides, nuclear hormone receptors are coming up or transcribed significantly in our data set. We curated all the nuclear hormone receptor in Ascaris. And the interesting thing is like the number of nuclear hormone receptors are also highly reduced in the parasitic nematodes as like chemosensation GPCS compared to the free living nematodes. And these nuclear hormone receptors are involved in like several functions like innate immunity, iron uptake, and lipid metabolisms, though the exact functions of nuclear hormone receptors is still unknown. But it's really interesting to see like in future, like are they really involved in any sort of sensations during the migrations. So in this chapter, to summarize, we saw that larvae are more migratory towards the liver, and we saw that fatty acid metabolism related genes, cilium formations, and chemotaxis related genes are upregulating, and we saw neuropeptide signaling pathway control the sensations during that process when they respond to the liver-stimulated larvae, and the ligand receptors activation happens as I saw the specific ligand neuropeptides and neuropeptide GPCS in this process. So we assume that in hepatometabolic migrations, they are really involved in this process. So then like we came to our next questions. How do larvae behave when they respond to liver and lung homogeneous? So we saw that like in molecular level, we see like genes are upregulating is coming up, but actually when they is come to the physically, what is happening? So to understand that things, to see the behavioral features of Ascaris, we have developed a microfluidic device. And based on that, we made our research aim as like fabrications of microfluidics and exploring the chemotactic responses of Ascaris soom larvae to pig liver lysate. So before going to this chapter, I want to tell about microfluidic systems and their involvement in the worm biology. So microfluidics is actually like small device that is like inert and like transparent and can be used to analyze the worm's behavior in a like tiny and small environment. And it has advantages over the traditional assay techniques, like the traditional assay is like more laborious. It just like handle the less sample size and they are more robust. And in C. elegans, microfluidics is being used for like several experiment or function for several experiments, like from imaging to metabolic studies, from the behavioral studies to worm scanning. So in C. elegans, it's really used like for different sort of functions, and the microfluidic device gave like lots of advantages, including they can use the screening of the genetic variants. The worms can be handled in a large amount, and it's easy to control the liquid in a small quantity into the device. So to do that, we come to know, we want to know like what is happening or what did people do like for C. elegans or other nematodes for understanding the behavior in the microfluidics arena. So in C. elegans, mostly that's the free living nematodes. They use the microfluidics for behavioral and cellular responses of worms. And it has been assayed for like different sort of gases, nutrients, and chemoattractant, and even for the like temperature sensations. And in the microfluidic device, anyone can like introduce the liquid chemicals in both spatial and the linear gradients. However, as I told, like this device is not like yet developed for the parasitic nematodes. Like for example, Ascaris, they need low oxygens or like homeothermic environment. So we have to modify the microfluidic device to understand the behavior of Ascaris into the microfluidic maze. So based on that thing, like as they are not available for the parasitic nematodes, we try to fabricate one microfluidic devices. Yep. So we fabricated a microwave device and characterized that device like compatible with the Ascaris larvae to move into the microfluidic maze. So this device is actually made and fabricated in Aramati University from our collaborator, and actually Peter did that. And then this constructed from the polydimethyl sulfoxane is like materials used to fabricate that devices, and to smoothly move the Ascaris larvae into the microfluidic maze, we characterize the gap size of the pillars. You can see around 40 micrometer to smoothly move. Smoothly move the larvae inside the microfluidic maze. And if you look at the basic structure of these microfluidic devices, you can see like they have like two inlets to make the linear gradients, and one worm inlet to insert the worms into the microfluidic maze, and there's a like outlet to release the fluids inside the maze. And this is the like typical structure of the microfluidic maze you can see here. And then like you can see like how stable the gradients are there. Like you can see how the gradients are shifting based on the height of the input channels. So it's really stable. I can see the video is moving. Yes. Yep. So this is a like outline of a microfluidic setup for which I have been done like for my experiment, but actually it looks like that. It's so messy. Anyway. So yep. So before going to do the behavioral analysis, first we have to characterize the locomotion of Ascaris inside the behavioral arena. C. elegans like showed like 4 basic locomotion behavior into the microfluidic mazes, and we observe the similar sort of behavior into the microfluidic maze, which are like forward movement, sudden pause, and the reverse, and the omega turn. This is the weird behavior of the C. elegans, that like they immediately detect something and they bend it and go through the to the chemicals or like attractant. If you can see the videos, you can see all of the movement of Ascaris larvae into the microfluidic maze, which shows all the forward movement, pause, reverse, and the omega turns. So they showed all the similar characteristics like in C. elegans. So that shows that like the movement of C. elegans is mostly conserved like the C. elegans, and you can see the trajectories of a single worms that moving from like start to in and how they are going from one point to another point. Okay, so we characterize the locomotions in Ascaris. Then we did the behavior analysis. For that like thing, we cultured the Ascaris larvae, and then like we take the motile larvae, and then we insert the motile larvae into the microfluidic maze, recorded the videos, and then we analyze those recorded video by software. So into the maze, what we did, like we input like around 15 to 25 worms and let them settle down for 60 seconds, and then like we recorded the video for next 120 seconds after initiating the gradients to make the linear gradients. So it's like typical videos. Okay, so how they are moving inside the microfluidic maze. So we take those videos and analyze these videos using the software. Anyway, so to understand the shifting of the microfluidics, understand the behavior of Ascaris towards the linear gradients of liver homogeneous or lung homogeneous. So what we did, like we fractioned the time of the 120 seconds to first initial 20 seconds, next 20 seconds, and 120 seconds. And we see the positions of x coordinates towards the gradients and how they are shifting towards the x coordinates, because on that positions like the worms are moving to the gradients. In case of RPMI, we see there is not much changes in the 20 to 60 seconds or 60 to 120 seconds, and these are non-significant changes. But when in case of liver homogeneous, they are significantly shifting towards the liver homogeneous. And in case of lung homogeneous, they also showed like non-significant shiftings towards the lung homogeneous. So all significance was calculated based on the x coordinates because we want to see how they are moving towards the x coordinates, which where the gradients are present. So anyway, the shifting is happening. So we want to know like that what is actually behavioral features they are showing during that movement. So we extracted those behavioral features of Ascaris larvae using like triapsid tracker. Sorry, it's like basically used for the C. elegans movement. We curated around like more than almost 3,000 features, but we only focused on 21, which are only related with the forward movements, pausing, reversing, and turning, which are related to the chemotaxis. And we saw that the features are actually like segregated based on the behavior, based on the chemicals like liver, lung, and RPMI. And the heat map shows the all Z score of the behaviors across the different time frame of different chemicals. Then actually want to see like how much larvae they are migrating. So in case of chemotaxis, we saw that in C. elegans, they move through a like shorter distance when they are going to the attractant. So you see like the larvae in the liver gradient, they are like taking the lesser path compared to the RPMI and the lung extract. And like when they are going to the liver gradients, they are mostly directionals. On the other case of RPMI and the lung, they are mostly like randomly moving like in the circle. So that's a trajectory plot shows that like how they are showing directional movements to the liver gradients. So then like we want to see like, is it actually chemotaxis? So we want to focus on like a few features like in C. elegans, they have like two features like roaming and dwelling. So in case of roaming, when they go from the lower concentration of food to the higher concentrations, so they actually show the behavior with the high speed and the less turning. We saw the exact behavior features of Ascaris sum larvae with the significant higher forward frequency compared to the RPMI, and then less turning. So we assume that is with aligned with the C. elegans, they have like similar sort of chemotaxis functions. So in overall, like what we see like in the macrofluidic maze, in case of RPMI, when they are initially going into the macrofluidic maze, they have a low speed with the infrequent turns, and by time in RPMI, they consistently keep the low speed and then they start to infrequent turns. That means they don't detect any chemicals or stimulant. But in case of RPMI, when they are initially in the microfluidic maze, they shows like high speed but frequent turning. But when they sense the liver gradient, they start with the consistently keep the speed, high speed with the infrequent turns and with the high forward frequency. So it overall says that like it shows some chemotactic behaviors when they are responding to the liver homogeneous. So in this chapter, like what we did, like we fabricated and characterized the macrofluidic chips, and we saw that they have a conserved locomotion behavior as like C. elegans, and that we saw larvae are shifted towards the liver gradients with the showing the similar pattern of roaming behavior like C. elegans with high speed and less turning, and they have like we saw like increased foraging speed, like I didn't discuss here anyway, but we see that. So overall, in summary of the thesis, what I did, like we curated the ortholog sensory genes in Ascaris zooms and characterized the sensory genes in tissue-specific transcriptomes and transcriptome of Ascaris larvae in vitro chemotaxis. We also fabricated and desired the channel size for the macrofluidic maze and confirmed the chemotaxis in Ascaris macrofluidics. And in future, we want to generate the 3D high resolutions of identify the individual neurons and then like develop the CRISPR RNA methods to analyze the gene functions, as we just assume that genes, but to prove the function of the genes, we need the functional genetics. And to develop the preserve the chemotaxis, we can do the laser evaluations, develop the anti-GPS antibodies or CRISPR-I. Finally, I want to acknowledge like there's a main part, like thanks Aaron for giving me such opportunity to work in these amazing projects. I learned a lot from that, and like we see something like different, like what people believe earlier. And thanks my co-supervisor Kasia Koshmanesh from RMIT for helping me with the microfluidics experiments. Thanks all the JX lab members Luis, Nijoy, Balu, Amrita, like they are really Achao. They're really amazing people helping all time with anything. And the other Jex Lab alumni like Joy, Sam, Swapnil, yeah, they are really helpful with my PhD journey. I like to thank my PhD committee, James, Robin, and Verena. And I like to thank my PhD deep course, Natalie and Hugh. You are really amazing for like a couple of years. I like to thank WEHI Education, Sarah. Like I just annoyed her every time for mine. This thing, that things that they are doing it. That's really good. Thanks, Peter from RMIT to developing the microcredit devices. Thanks, Melbourne Research Scholarship and WEHI for supporting with the scholarship. And one thanks to give to the Diamond Valley folk who always give the adult worms for the eggs and the freshly slaughtered liver and lung. Otherwise, I can't do my PhD. Yep. So my collaborators from around the world and many, many others. And thanks to all my friends and family who are really supporting me last couple of years, like really amazing journey. And thanks all joining today. And I'm happy to take any questions or suggestions.
When you ask a question, can I please wait until I give you the microphone, because we do have people joining us online, and otherwise they won't be able to hear. So, and then I don't see the chat, or because I don't think Pradip can, just to let us know if there's anyone asking a question. Oh, is it on? Hello. Hello. All right, that's gonna be different. All right, what we'll have to do is say, unfortunately, sorry for those of us joining online, unfortunately our podium or our little handheld mics aren't working. So what we'll do is I'll ask the audience member to give a question. Before you answer the question, if you could repeat it so the people who are joining us online can hear it. Sorry about that.
Nice.
Thank you so much. This is really and my only suggestion is next time more. Okay, so I have two questions. So one is you mentioned iron sensing.
Yes.
So which receptors is the parasite pushing?
For iron sensing, I'm not sure, but I think not is like chemotaxis process. But yeah, I'm not sure like which specific genes are like involved in ion sensing. But if it is like a chemotaxis, there might be some other chemosensory genes might be involved to take the iron sensing. Like most of the downstream signaling genes are like guanylate cyclase or CNG receptor channels. They are really involved in the chemotaxis process. So, but I'm not like exactly in which one. Yes.
So that's a lot of issues, a lot of information and immunity that it has to be able to invade. How concerned would you be if you stop this migration? Then you'd also stop the ability to avoid that immune response, and so you'd be invading it.
Yep. So Ricardo asked questions about like how could it block the like immune response, or how could they block the?
So would the parasite need all the sensory machinery active to be able also not to kill the host?
Yes. So the main point is about evading the immune system of the host. So actually it's focused on like mostly depend on the secretory proteins. So they release like huge amount of secretory proteins when they are migrating, unlike come to the even like the intestine. But I think they then modulate the host lymphocyte granulocytes a little bit, and they puzzle them. So that's how they evade. But when we assume like if we can block the chemotaxis somehow by perturbing the chemosensations, they might be susceptible for the host immunity and they might be killed there. Like because if they stay there for a long time, like in the liver, because that's why they just like as quick as possible, they evade the liver, come back to lung, and then also they evade the lung and come to the intestine. And in the intestine, there is like, for the adult, like they have a like matured like immune system, but for that's why like children are the most vulnerable for these things. So in the adolescence, like children can't protect this thing, and then the Ascaris grows. But mostly like if you can block them in the initial phases, the host immune process can kill them inside the tissues, and they can't complete their life cycle. Because and also like in the process, like they also lose lots of like, it's like costly. So like when they go to the liver, they also like, you can see the like some spot in the liver. So that's actually killing the larvae, and they make the spot there by the host immunity.
Any other questions? Yep, Julie.
Yes. Yes.
Movement, or is that more like what they use to process their food, or how is cilia involved in?
Okay. So Julie asked about like how cilia works in the larvae, and are they related with the movement or something? Yep. So yeah, they don't have actually cilia like the bacteria. So it's actually cilia in nematodes are like the neurons. So when they sense some sort of chemicals, they have to like reshape their cilium, and they senses those signals from the environment, and by this how they move. Like they saw something, sense something, and then cilium detect that, change their structure, and they moved towards that. And it's shown that in C. ligands, if there any defect in the cilium structure, they lose their movement or like sensation capability. So that's really for the nematodes, for the movement or locomotions. Yeah, the common one is like they're using C. elegans, but the problem with that C. elegans is like it's a free living nematodes. So it's not actually mimic like the parasitic nematodes. So yeah, people are still using the C. elegans, but still we have to work on the parasitic nematodes specifically to see that how it's changing or happening. Okay, that's interesting questions. So actually, like previously, people thought like, oh, they will not go to the liver, they can go to the lung. Why didn't go? We don't know that. And previously, like even like they inject the larvae into the other typical site like lymph nodes, but they came back again to the liver and then go to the lung. So it's not like that. It's random. They are anywhere. They are coming to the liver. So that's why we focus on the liver. That something is happening in liver, and they are releasing some cues or like any signaling, and then they migrate and complete their life cycle. And without that completion of the life cycles, they can't survive and they can't become mature. So yeah, it's like it's still not unknown why they are coming, but we are trying to solve it. Like, is there anything connected to the chemotaxis?
I apologize because we did start a bit late, but I'd like to thank you all for joining. Please join me again in congratulating Pradip on his really excellent thesis work. I hope you enjoyed the presentation. With that, I'd like to again thank Pradip for an excellent piece of work and congratulate him on his thesis studies.
Thank you all.